Hi there, welcome to lesson 7.16. In today's lesson, we'll look at nematodes as indicators of soil health, the different measures that are being employed when using nematodes as an indicator of soil health, and we'll also look at how soil structure really affects the population dynamics of soil nematodes. So first of all, let's just go back to a slide you would have seen in a previous lesson where the authors have studied the role of nematodes in soil food webs by seeing how nematodes enhance plant growth and nutrient uptake under carbon and nitrogen-rich conditions. And they found that nitrogen and phosphorus were increased as well as plant biomass as a result of all the different nematode groups playing a part in the soil food web and affecting that soil food web and therefore plant growth. So first of all, let's look at root herbivory. So root-feeding nematodes. When these guys feed on the plant roots, they actually release root exudates, and that increases exudation. And this in turn stimulates fungal and bacterial growth as fungi and bacteria then consume the exudates that come out of the plant as a result of the herbivory. So this enhanced microbial community then immobilizes nitrogen and phosphorus from the bioavailable pool and from the fresh organic and the soil organic matter that is present in the soil. They basically take those nutrients, that nitrogen and phosphorus, into their own cells. This stimulates then the bacterivores, fungivores, and omnivores, so bacterial feeding and fungal feeding nematodes. Their numbers increase as a result of this increased biomass of bacteria and fungi. These guys then, as they consume bacteria and fungi, excrete the excess nitrogen and phosphorus that the bacteria and fungi provide, and they unlock these nutrients from that fungal and bacterial biomass and allow this to enter the soil, which is then available once again to the plant, as well as to feed more fungi and bacteria. Fresh organic matter that actually has been added in the study, but this can also be seen as leaf fall or root shedding or any organic matter that enters the soil and that is fresh, further stimulates the growth of bacteria and fungi because these guys then start to consume this fresh organic matter. So this further stimulates microbial biomass and activity, and this is similar to how exudates function as well. And of course, the soil organic matter as well is being continually munched on by bacteria and fungi and further transformed. And so in totality, nematodes increase plant nutrient availability and uptake and therefore increase plant biomass. So these are the roles and the functions of the various feeding groups of nematodes in the soil food web and in the soil ecosystem. In addition, nematodes can be used as a very important soil health indicator. All biological indicators, so all creatures within an ecosystem, can be used in order to assess the health of the ecosystem. That's because a lot of these creatures, particularly little ones like nematodes, are sensitive to changes in disturbances, or in the case of agricultural soils, management of the soil. They correlate well with beneficial soil functions because they play these vital roles, key roles in the soil ecosystem. And therefore they can help to explain the ecosystem processes. And so that means what is happening in terms of the available food to the different nematodes, or the predators of the nematodes, what is happening to them as a result of nematode abundance or nematode species that are living in that ecosystem. And this tool, using nematodes as soil health indicators, the data from that can then be used to give comprehensible and useful information to land managers so that they can make some decisions about further improving soil health based on the nematode data that is gained from that soil ecosystem. There are very many good reasons as to why nematodes are great soil health indicators. And that includes the fact that they are the most abundant and diverse soil mesofauna, and therefore key indicators of soil quality and health. We also understand the taxonomy and feeding habits of nematodes. They also occupy primary, secondary, and tertiary consumer roles. So primary being the root-feeding nematodes or herbivorous nematodes, and then the microbial nematodes and also predatory nematodes. Free-living nematodes indicate soil biodiversity and disturbance. So if there is a biodiverse community of free-living nematodes, it's a good sign. And when disturbance happens, these nematodes often disappear very rapidly, particularly the fungal feeders, the omnivores, and the predators. In general, diverse community of nematodes increases plant growth, and this alters microbial community as the plant exudates and the organic matter from the plants feed the microbial community and therefore the nematodes. Nematode community changes is a reflection of soil disturbance as well, so the different feeding groups and the diversity within those feeding groups, as well as the composition of that community. So if one wants to assess the ecosystem health and how disturbed it is, you can look at the nematode community composition. C. elegans, for example, can be used for a rapid chemical toxicity assessment of pollutants. There is reduced growth of the nematode and its reproduction is affected. Offspring counting assays can be used in several heavy metal ecotoxicity evaluations. So this is just a snapshot of how nematodes and the nematode community in a soil can be used as a soil health indicator. Nematode community and diversity characteristics can be used as indicators, and this is influenced by vegetation type, geographic location, climatic and soil conditions. Size of water-filled pores is a major structural limitation, and therefore it affects the community and diversity of nematodes within the soil. Some of the characteristics that can be measured within the nematode community include abundance of all nematodes, the diversity, the different species and the different families that nematodes are classified into, the structure of the community, the composition of what kinds of nematodes are present within that community. So are there bacterial feeding nematodes and that's all we've got, or are there also predators, are there fungal feeders, and so on. And also the metabolic footprints can be measured, and this is the measurement of the metabolites that result from the livelihoods of particular organisms, in this case nematodes. So what is excreted via their metabolism into the soil that can actually be measured to understand the composition of what kinds of nematodes are present within the nematode community. Species and community measures include the number of taxa, the absolute abundances, trophic structure, so the different feeding groups based on relative abundances. Obviously, soil conditions determine nematode community size, complexity, structure, and the metabolic footprint. And these are just some examples of nematode indices that can be used, taken from this particular study in 2009, that they used in order to assess the nematode community composition within the soil. So the nematode indices that they used include the Simpson's Index of Dominance, and this is just to give you examples of what these indices look like and what they measure. So the conditions that the Simpson's Index of Dominance describes is family diversity and dominance. Larger values indicate high family diversity, and smaller values indicate community dominated by few families. Fungivore to microbivore ratio, or fungal feeders to bacterial feeders ratio, demonstrates the decomposition pathway. So how is organic matter decomposed? Is it decomposed by bacteria or is it decomposed by fungi, depending on that ratio of fungivores to bacterivores? Larger values indicate greater fungivore dominance. Total maturity index describes the successional stage of the community. and ranges from rapid colonizers all the way to persisters, which dominate the community. So when there's a disturbance, oftentimes the nematodes that are the first to come up are the ones that rapidly colonize bacterial feeders that feed on the bacteria that are the most predominant in that soil, and then these larger omnivorous predator type of nematodes are the persisters that, if the soil disturbance is minimal, they will stay in that soil for long periods of time. So the presence of persisters indicates a stable ecosystem, as these nematodes are characterized by long lifespans, low reproduction rates, and are highly sensitive to disturbance. There's also the enrichment index, which is the availability of nutrients following disturbance, and that ranges from all the way from low to high nutrient availability. There's also the structure index, and that is a measure of the connectedness of a nematode community, and that ranges from few connections to many trophic links or feeding links within the soil food web. The body size of nematodes is also a tool that can be used, because disturbance actually reduces the larger bodied organisms. So oftentimes when the soil is plowed, for example, a lot, it may completely remove predatory and omnivorous nematodes, the larger nematodes. Body size changes metrics is another popular assessment of community structure changes in response to environmental changes. And in general, body size-based assessments can actually reflect all kinds of disturbances, including clear-cutting, aridity, fertilizer applications. Any disturbance to the soil ecosystems, any changes can be assessed as to their severity based on this body size assessment of soil nematodes. So as I've mentioned before, nematode communities and soil structure are very interrelated, because soil porosity has a primary effect on nematode community assemblages. Intra-aggregate architecture, so that is the spaces between the aggregates, determines the grazing microhabitats for nematodes. They live in thin water films of soil pores that are between 30 and 90 micrometers in diameter. So this is very specific, and when the structure of the soil is such that it allows for these spaces for nematodes to live in, that is a very good indicator that that soil is healthy, because it allows for these water films which nematodes require to live in. Increased organic matter, such as compost or manure, increases soil porosity to around 100 micrometer pore sizes, and this leads to an increase in nematode abundance. Pore sizes of 200 to 1000 micrometers are greater than the largest nematode diameter, and this reduces colonization and habitation in macroaggregates. So there is a fine line in the soil structure as to whether nematodes can live within that soil, because the larger the diameters of the pores and the spaces within the soil, the less moisture availability there is for the nematodes, less places to hide from predators. And the bottom line is that these organisms really require specific habitat to live in, and so therefore soil structure is paramount to their existence within the soil ecosystem. An interesting study came out in 2006 where the authors measured the nematode distribution in soil fractions. So they divided the soil into various size fractions and found that over 50% of the bacterial feeding nematodes were found in interaggregate pores and over 20% in fresh organic matter. So this is where the bacterial feeders were most likely to be found. Less than 25% were found in aggregates of more than 200 micrometers in diameter. And remember, these are not pores, these are actually aggregate sizes, as opposed to pores that we spoke about in the previous slide. Similar distribution was found for the fungal feeding nematodes. However, herbivorous nematodes or root-feeding nematodes. Majority of those were found actually in aggregates that were greater than 200 micrometers in diameter, and over 35% in interaggregate pores. The predators and omnivores were almost entirely localized within the interaggregate pores, and only very few were found in association with fresh organic matter, and around 25% of predators were actually within the aggregates that were greater than 200 micrometers in diameter. And nematodes did not have entry or access to the microbial population in pores that were between 10 and 50 micrometers in diameter. So this is just to show us the kind of habitat that the various nematode trophic groups like to hang out in, the sizes of the aggregates that are required in order for these organisms to be able to have their lives in. So we have come to the end of lesson 7.16, and our last lesson on nematodes will focus on how soil management affects nematode communities. So you'll be hearing from me again then. Bye for now.